All right, so this picture here sort of introduces transcription, which is what we're going to mostly focus on today. And transcription doesn't happen, it's going on all the time in the cell. It's different from replication in that replication is only happening when the cell is getting ready to divide. So all the DNA needs to be copied, and it only happens in S phase of the cell cycle before mitosis. These two processes that we're going to talk about in chapter 17, transcription and then translation, they are going on continuously in all cells all the time. Different cells are transcribing different portions of the DNA. And this is to remind you, I just put the picture up that has the eukaryotic cell to remind you that the transcription is going to be happening inside the nucleus. And then that second part, the translation, which we'll talk about on Friday, is happening outside of the nucleus, literally on the endoplasmic reticulum, in ribosomes. This is to try to give you another little bit of context for this. So here's the double-stranded DNA molecule like we were looking at in the last chapter. And they've labeled some genes, different colors. And if you want to think about that as far as like when we were talking about Mendel, and maybe gene 1 is the gene for flower color, maybe gene 2 is plant height, maybe gene 3 is pod shape. And each of those genes, which can be inherited from one generation to the next, they are comprised of a particular sequence of DNA. And then to turn that DNA into the protein that actually gives the plant the color of the flowers, the height, in order to do that, you need the transcription and the translation process. And as I said, today we're going to just focus on transcription. That's just bigger. I put this in to remind us that the DNA is complementary, right? Double-stranded and complementary. And notice that the RNA is single-stranded, and there are some differences here. I'm going to stop sharing this screen and share a different screen with you where I can actually draw. Let's see if I can just put up the whiteboard here for a second. So for RNA, which is going to be the product of transcription, there are a couple of differences in the structure. And one difference is we have a different sugar. So we have ribose now instead of deoxyribose in the nucleotides. There are no thymines in RNA, in any of the kinds of RNA. So I'll put a big slash through that. Thymine is replaced in RNA with, we call it another nucleotide, uracil. Like thymine, it is a pyrimidine. So it has one ring, but it is a different, little bit different nitrogenous base. And the third feature, the phosphate's the same. Still has the phosphate group attached to it to make up the nucleotide, right? So RNA nucleotides are going to have ribose instead of deoxyribose. There will be no T's in them. So what the complementary, so if a sequence of DNA, for example, if the DNA sequence says A, right? If there's an adenine there, in the RNA, when we do the transcription, we would put complementary to that A, not a T, but a U for the uracil. Uracil does not substitute for both A's and T's though, right? So this is a common mistake that students make. They want to, when the DNA has a T in it or a thymine, they want to put a U here too, but RNA still has A's in it, still has adenine. So when the DNA has an A or an adenine, the complementary RNA is going to have uracil, but when the DNA has a T, complementary RNA nucleotide is still going to be adenine, okay? There are lots of different types of RNA. We are only going to talk about 3 types. Literally, in eukaryotic cells, there are like 12 types. Come to genetics, learn more about it, right? But we're just going to talk about 3. messenger RNA, ribosomal RNA, and transfer RNA. Okay. I'm going to draw pictures of those or tell you a little bit more about those in a minute after we go through the actual transcription process. So let's get rid of this and go back to this. So what you need for actually the process of transcription, there's 3 parts to it. The first part is initiation, second part is elongation, and the third part is termination. So, and it's just like the words actually sound like. Initiation is getting the process started. Elongation is adding nucleotides, and termination is ending the process when you get to the end of a gene. So like when you look here at this where it's got gene 1, gene 2, gene 3, these are finite amounts of DNA. We are not going to make a transcript, a messenger RNA transcript of all of these. We are just making a copy of one gene at a time. Okay. So here is the beginning part, the initiation, and I put on the slide that one of the main things that's required so that this enzyme, this gray thing here is the RNA polymerase enzyme. How it knows where a gene begins is this particular DNA sequence. We call it the top box because it's TATAAA. I think I put 1 too many A's here. It's just 3. That sequence is what RNA polymerase uses to find the beginning of a gene. When it finds that beginning, it binds to the DNA and all by itself, doesn't need helicase, doesn't need single-stranded binding proteins. It's just going to open up a small part of the DNA to begin elongation. And I'll show you this picture next. This is what we call the transcription bubble. So here's the DNA open. See the blue strands are the DNA. And here's the gray RNA polymerase, and it's adding bases. Just like DNA polymerase, it's going to add bases 5' to 3'. So it's actually, notice it's reading this strand, the 3 to 5 strand. We call the 3' to 5' strand the template strand. Or sometimes you'll see it called the coding strand. And then the complementary piece of DNA is called the non-template or non-coding strand because it is not going to be transcribed into message or into RNA. And you'll see the base pairing here that we talked about on the little whiteboard where I said if the DNA has a thymine, RNA is going to put in an adenine, but if the DNA has an adenine, the RNA complementary base is going to be uracil. Notice they've made the RNA red. It's supposed to help you see the difference. And notice we're not copying both strands. We're just copying the one on the bottom here. This is the elongation phase. RNA polymerase is adding bases super quickly, and it is also does not proofread like DNA polymerase. So it might make mistakes. I would like you to be muted. Okay, so this is that same slide that we started with here. It's just added the elongation phase and the termination phase to it. So initiation, we need the top box in order to get the RNA polymerase in the right spot. RNA polymerase joins the DNA and opens it up and starts adding bases. That's the elongation phase, right? So we've got 5' to 3' here. RNA polymerase, notice the red arrow, it's going this direction. And as it keeps moving, it's going to keep adding bases. This transcript is going to get longer and longer. The newly synthesized RNA, no matter what kind it is, messenger, transfer, or ribosomal RNA, we always just call it the transcript. And then the termination phase, simply, you can think of it like the initiation phase. There's a particular sequence of bases that tells RNA polymerase this gene's over, time to let go. Now, as I mentioned, there's 3 different types of RNA that have to be made by the cell all the time. And those types would be messenger RNA, which this is a picture of. It is always single-stranded. It's usually about 300 nucleotides long. There is also ribosomal RNA, which I don't have a picture of, but ribosomal RNA. Ribosomal RNA, like I said, I don't have a photograph of that. It's usually also single-stranded, and I'll show you a real quick picture of transfer RNA. It looks like this. It starts out as single-stranded, but then it forms these hairpin loops, and this is going to be important structure so that it can actually do the translation, which is the second half of the chapter. So we've got the elongation phase and then the termination phase, and now we're going to focus on some of the events that happen to this completed messenger RNA transcript before it can be put out into the cell for translation. So if we zip back to this first picture, see how this is really long? We call that the pre-RNA. That has all of this sequence, all of this gene in it. Notice that the next bar says RNA processing, and notice that after that happens, the RNA is much smaller. And it has different colors on the ends. There are 3 types of RNA processing in eukaryotic cells. And we are just I'm going to show you a picture of them, give you definitions, and that's it. The RNA must be processed before it can leave the nucleus. Okay, so we would call this one the pre-RNA and then this one the mature RNA. Okay, so let's go back and look at what those 3 events are for processing. They're titled post-transcriptional modifications because they're chemical changes to the RNA after transcription has happened. This slide shows you the first 2 out of the 3, and then the third one has a slide all by itself. So this slide shows the first one that I listed on your handout, which is the 5' cap. And that means at the beginning of the messenger RNA, the 5' end, after it is finished being made, there is an enzyme that adds a methylated guanine. And it is like, you can just think of it like putting a hat on, okay? It helps the RNA be correctly identified when it leaves the nucleus as RNA from this cell. And it also helps the RNA get positioned correctly in the ribosome so it can be made into protein, so it can be translated into protein. The other post-transcriptional modification that's on this particular picture is here. It's called a poly-A tail because it is a long string of adenines. And on this slide, it tells you specifically 50 to 250 adenines at the 3' end. And the function of that is also for stability, so that the RNA doesn't accidentally get digested when it leaves the nucleus. And the last post-transcriptional modification is this one, and this is called splicing. Now, most genes, when they're made in this pre-messenger RNA form, there are parts of them that are actually going to be used to code for the protein. And then there are sometimes short and sometimes really long sequences that don't code for the protein. They don't show up in the protein at all. Those pieces are called introns, and an intron means it's short for intervening sequence. Those have to be removed before the message can leave the nucleus. Notice how long this message is, this piece of RNA is right now, right? And then you can see in the darker orange color, they've put all of the pieces that are essential for this gene to be turned into a protein here. Those portions are called exons. And we're going to put those exons together and cut the introns out. There's the 5' cap. There's the poly-A tail. Okay. Now this is a mature messenger RNA, and it can leave the nucleus and go to the ribosome to be translated into protein. And that, I think, is the end of what